Bretschneider from Warwick System Biology Center. Please.
Thanks very much, Sasha, and many thanks for inviting me to that really exciting meeting. It's a great honor to be here. So we heard this morning two talks about two very different cell types. So gliding fish keratocytes, okay, and then mesenchymal cells that sit in the matrix and pull. So I'm going to talk about Dictyostelium, and Hans already gave a very nice introduction into the biology, so I can skip most of that. I want to concentrate on a couple of things. The first one are actin waves, and again Hans already presented even a model for that, so which I'm not going to do, but I would like to point out some details that are very interesting in terms of mathematical modeling. And then I would like to speak about a couple of experiments that help you to actually separate what is happening at the front and at the rear of cells. So in terms of mathematics, what we would like to learn here is, so how can we disentangle different time and space scales. And this is a point that came up in the discussion before. So the microscopists would like to actually bring together images with mathematical models, and I sit somewhere in between. So I'm very fond of video microscopy, and actually also would like to be able to match up what we see in the model with image data. And so I'm covering two different aspects here. So one is fitting models for cell orientation, and again we had a discussion on how simple needs a model to be, or can it be, and you will see we're talking about very, very simple models here. And the last one is again a different aspect that relates to cell mechanics, and that is a biophysical model for, not for genuine blabbing, but only for bleb site selection. Okay, so this is a collaboration with Rob Kay here in Cambridge, and the earlier works was all done in Günter Gerish's lab when I was at the Max Planck in Munich. So you've seen quite a few movies about moving cells. So Dictyostelium is a amoeboid type of cell, and here you see it chasing the tip of the pipette, which is filled with cyclic AMP. And so you already heard so that this plays a fundamental role in the life cycle of Dictyostelium, because Dictyostelium cells, when being starved, secrete cyclic AMP themselves and use that as a mechanism to aggregate and eventually come together as a almost multicellular organism that consists of individual cells, and that organism even can crawl in form of a slug and eventually culminate to produce a fruiting body with spores. And from these phase contrast images, so you see as soon as cells start responding, they become highly elongated. If we look at a fluorescence image of actin and myosin, so we see that the cell forms two functionally very distinct ends. Okay, so here we have a marker in, maybe we can close that shade here, so it's a bit too bright. Thank you. So here we have a marker for actin in red, and And so you see the cell front lights up in red, and this is where actin polymerizes into a highly branched, dense network, and the polymerization of that network provides the mechanical force to actually push the cell front forward, whereas myosin is enriched in the back of the cell, where its contractile mode of action is responsible for retracting the cell rear. And from an engineering point of view, you would really like to have a simple picture like that, okay, where we can say we have a modular system where cyclic AMP binds to receptors, okay, and this is coupled through a G protein network through a box which takes care of the signal transduction, and then we have as an output actin polymerization. And then you see here's already a slight complication, because there's also an arrow going back into the signal transduction, because actually there's many components that bind to the growing actin network and reinforce the signal there. And then the polarity network essentially describes the formation of the front-rear axis, and in particular the myosin accumulation at the rear. If you look at the molecular level, this becomes very messy, okay? And this is most often where mathematicians are thrown and go into sleep mode, okay, because they think, okay, he's going to talk through all these individual molecules and probably never stop. But I try to convince you so that the main task I think mathematicians have is helping the biologists to disentangle that. Because what is obvious is, so this is a network, but we do not capture any spatial or temporal dynamics here, okay? And essentially, these processes are very much separated. For example, you have that branch here, which results in myosin-mediated contraction. And we've seen, so that takes place at the back of the cell, okay, not at the front. And one key element is that area here shaded in yellow, which is actually responsible for localizing actin polymerization at the membrane, okay? So we have these phospholipids here, which are constituents of the cell membrane, and specific proteins with specific binding domains can bind to these phospholipids. So the idea is so that in response to a cyclic AMP signal, we have a signaling cascade which results in local enrichment of PRP3, and then other molecules can read out that signal, and further downstream the ARP23 complex will result in actin polymerization. Having said that, so this is the picture which used to be the dogma for many, many years, and as you can imagine, things are slightly more complex. And it is actually such so that Dictyostelium cells, in which all PR3 kinase signaling has been abolished, are still able to chemotex. So meaning you can do without all this signaling module here. Yeah? Also, what is upstream of this signaling module is the so-called RAS signal transduction box with RAS C and G playing a prominent role in dictyostelium. And you can show so that even cells without these can chemotex, for example, to folate. Another recent finding is so that there's actually a direct way which circumvents all this PRP3 kind of signaling box where actin can be polymerized by a factor called ELMO-E, which is a protein that binds to G proteins that are directly coupled to the cyclic AMP receptor. Okay, and the understanding is so that it is actually the experimental system which made people believe for decades so that this RAS and PR3 kinase signaling model plays a much more prominent role than it does. Okay, and we now think so that this module is has a specific important role in a process called macropinocytosis, which is simply cellular drinking, so where cells take up fluid. And I hope this is going to play. Yeah, so you see here the uptake of fluid, which is then internalized. Okay, and you can actually show. So these are laboratory strains that grow axenically. That means they grow in liquid medium. Okay, and they have been mutated such that it's easier to keep them in the lab and grow them in liquid culture. And you can show that actually one single mutation that inhibits this RAS pathway is enough to create this axenic phenotype. Yeah, so the picture is such so that we need to consider this module here in parallel with other actin polymerization pathways. Nevertheless, so this is very interesting because you can learn a lot about the actin polymerization machinery going on in waves. So here we look at the cortex of a dictyostelium cells that shows very different patterns. And so this is a pseudo-colored image where you merge two subsequent frames of a sequence, so one colored in red, the other in yellow. So whatever appears in yellow is stationary, and we have elements in green that disappear between frames.
Question, please.
Oh, yeah, sorry, I've got a very poor vision, so you need to shout or just...
Can you say what's the labeling, the GFP labeling? This lim E delta code?
Yeah, that's a lim E delta code. So that is a domain of an actin binding protein that is specifically labeling filamentous actin. Okay.
Like LifeAct, basically.
It is very similar to LifeAct, yeah, in what it stains. Okay. But people believe so that LifeAct probably is a better marker than LemE. Yeah? And so if I play that movie, so you see that this cortex. So this is image at the very bottom surface of the cell is highly dynamic, and so these network structures are produced by the action of formins, so which forms bundles. Okay, and then we have these patches here inserted, and
May I ask something?
yes.
Does it really give you any advantage of doing it as an overlay rather than just subtraction? If you subtract one from another, you would see again something that moves.
That is right. I mean, so we use subtraction to actually measure speeds of these bundles growing. Yeah.
But I mean, visually, you would say that that's give any advantage. I'm just wondering because I used to do subtraction, so that's why I'm just wondering.
Visually, you see from one image what percentage of the network stays stationary. Okay, so that's it's not a big deal, so you can do it both. What's happening here?
I hope it's not my question.
That would be advanced computing.
That looks terrible.
It's pretty anyways.
Okay, I better skip that. So what I wanted to show, so there was a small region where you could see a wave emerging. And so these waves can be seen in cells under normal conditions, but as Hans has pointed out, so a very nice experimental system is to actually depolymerize the actin cortex completely by using latrunculin A, and then you obtain cells that are void of F-actin and completely round. And then if you wash out the drug, after a while, so you see patches emerging and then these massive waves. And I have dug out this movie from a presentation I gave probably 10 years ago, but I think it's really worth seeing it if it plays. Something seems to be wrong. Come and see me in the break, and I try and play that to you. And yeah, so I'm only going to give a few details about the experiment. So we used photo bleaching of these waves and using double constructs to actually so you saw bleaching here, to actually find out which proteins will bind in which temporal order to these waves. And then we did a lot of 3D imaging to dissect the structure of these waves, and that resulted in this schematic drawing, so where we have the cell membrane here. And so one important component are myosin 1 molecules that can anchor the growing network or growing filaments to the membrane, and at the same time via an adapter protein called Carmel can recruit the ARP23 complex to the region of a growing wave. And then we have other factors like coronin, which are responsible for degrading filaments. And more recently, a very nice experiment was done by Carsten Beter and Günter Gerisch, where they used giant cells electrofused, and then this brings us back to what Sam said, so that you should try and change the physics of your model. And you think, changing the size of a cell, so that's probably impossible, but you can do it either by using electrofusion or by preventing cell division. And then what you find is quite a striking thing. So you see propagating waves here in red, so the actin marker, and then at some distance behind, you see in green myosin assembling. Okay, and the scale here is so that this scale bar, which is 10 microns, so roughly about the size of a normal cell, and you see how gigantic these cells really are. And so it's only because you change the spatial dimension of your system that you're now able to see, so that actually there is not only the components of a cell front found in that wave, but after that wave has passed, similarly the components responsible for forming the cell back are recruited. Okay. To sum up that part on the waves, so we can say these originate from PIP3 patches and resemble frustrated attempts of phagocytosis. However, so they more recently were shown to have a functional role in detecting substrate structures and moving in structured environments. So I have no time to go into that. So the molecular mechanism is very similar to what we find at the leading edge in Dictyostelium. So this is why they're a really interesting model system where we understand how most of the components are recruited. And then I've shown so that in giant cells, so we see there's a separation of front and rear pathways. And similarly, that can be shown in cells that are exposed to a uniform stimulation with cyclic AMP. So here you're looking at cells sitting in a flow chamber, and again that is a marker for actin, and then cyclic AMP is flushed over these cells now, and you see this cortical enrichment, which we call the Cringe reaction, and Hans again mentioned that yesterday, and again had even a model for that. I don't have one. But the interesting thing here is so that again using the experimental system where we always take cells labeled with one reference marker, in that case this LIM protein which decorates F-actin, and then other proteins, we can look at the recruitment of different components to the cell membrane. Okay, and so here we see LIM and then actin, and actually we see so that LIM is a good marker for F-actin because it shows up almost as the very first thing, and then here in this experiment, the cell goes to a stage where it's first all front, okay, and after a while we see that myosin is being recruited. So we see a very fast adaptation which dies down where the cell first is all front and then it becomes all back. So interestingly here the proteins which are usually thought to trigger actin polymerization actually show up rather late. So this is summarized here. So where in response here to a cyclic AMP simulated front, we see all the front components, showing up within 10 seconds, okay, and then very fast adaptation, and then afterwards the cell turns into a state where it's all back and decorated with myosin. Yeah. I would like to change topics here a bit, and
before you change, can I ask something? So in this, you showed this very interesting image with actin wave followed by myosin wave. So how is this related to the change in shape? So when the actin wave reaches the boundary, you have protrusion, and then myosin trigger retraction, or because you showed static in here.
Yes.
So what's happening with the cell, or it doesn't move much?
In that case, I must admit I do not know exactly what the interaction of the wave with the boundary is. Okay, so sometimes they stall. So in small cells, so the wave can actually act like a leading edge and then push out the membrane. I'm not sure whether this can happen here.
And I know about actin. I was most interested in what myosin.
It's an excellent question. Yeah. Any more questions on that part? Yeah. So the main theme here of the workshop is PDEs and evolving boundaries. Okay. So I thought I have to put something in, and there's actually quite a few groups who have worked on that problem, and combined models for cell orientation with models for motility, and actually looking at emergent behavior of cell motility and how that is driven by the chemical signaling. And interestingly, most people have simply taken the very first model in the field by Meinhardt, which is a 3 variable model, okay, where you have a signal and that results in autocatalytic production of an activator variable, and then you have one global inhibitor and one local inhibitor. And this is, for example, work by Charlie Elliott and Schicker and Catamaran and Björn Stinner, who are all here, where they look at the evolving boundary where the activator variable is coupled to this boundary and can result in pushing out that boundary. Okay, and the boundary itself is given by a force balance equation. Okay, where you assume isotropic pressure inside the cell, and you have viscous friction, and then intrinsic forces like membrane tension and resistance to bending. And one very simple question I wanted to ask is, so actually how good is that Meinhardt model? And that goes back to a very nice paper by Lea Edelstein-Keschet, where they compared different computational models, and I was asking, so yeah, could you, using image data, show which models performs better or can be ruled out or whatever? So we took 3 models. So the Meinhardt model, one by Levchenko and Iglesias, which is coming from a perfect engineering angle, and one by Otsuchi et al., which is based on the idea so that you have a signaling component that can either be in an activated state or an inactivated state. And so the actual nature of these models here does not really play a role. So we wanted to take simple enough models. And the experiment I'm considering here is again dictyostelium cells, but now responding to mechanical shear flow. So they sit here in a little flow chamber, and you have a pump system, and you can see a cell here that is first unpolarized, and now the flow is being switched off, so it elongates and then happily migrates steadily for 10 minutes or so. And we had older experiments where we exposed cells to a reversal in shear flow. So what you see here in green is again F-actin, and so these cells form a front protruding against the flow. Okay, you see if you reverse the flow, it takes a time for the old front to decay, and then a new front forms. And we took this as an experimental model to try and fit these different mathematical models to.
A question?
Yes.
So are these relatively early stage cells, so there's no real intrinsic polarization?
These are vegetative cells, yes. So what we do is, so we use some image analysis to extract the fluorescence here in the cortex, and this is plotted here as a time-space plot. So this is the coordinate along the contour here, and this is time. And this plot is for a single cell, and to start out, we used averages over a number of cells, okay? And then what we do is, so we fit, for example, the Meinhardt model here to the entire spatio-temporal process, okay? So here you see individual time points, but we fit to the entire sequence, and this is done in a very simple way. So we actually use actin as a readout for the activator. You can argue whether this is good or bad. And then, so we have a 1D PDE model on the cell contour, and we transform that PDE model into a system of coupled ODEs, which can be fitted very easily using very standard methods. So not much exciting here. So what you see here is now the integrated fluorescence intensity in a new front forming, okay, and in the old front which is decaying for 3 different conditions. So a high shear stress, a low shear stress, and the last condition is where we have a polarized cell and then simply switch off the flow. And here are the corresponding model fits. So you see, so this exponential decay is captured quite well by all of the models. And then there is quite subtle differences. For example, here in the low shear stress, so you see that the new front almost immediately comes on, whereas is quite a delay in the high shear stress. Okay, which is also captured by some of the models well. So we discarded the Otsuchi model at this stage. And if you now look at the individual parameters, well, one question is, so how do you obtain a unique solution? Okay, so how identifiable is your problem? So what we did is, so here we used a very simply profile likelihood estimation approach, where we plot for each of these parameters the goodness of fit, so the chi-square value for each model when we vary the parameter. Okay, in the ideal case, you obtain a parabolic shape with a clear-cut optimum, but you see here's a case, for example, for one of the parameters where you can change the parameter, but the goodness of fit doesn't change at all. So it's a in that case a practically non-identifiable parameter. We can fix that in case of the Meinhardt model very easily by realizing so that this inhibitor B actually is almost always constant, and so we take it out of the equation and actually replace it by a function of the fluid, the shear flow, and reduce it to a two variable model, and that is in the end fully identifiable. So we have three new parameters, okay, and you see the goodness of fit is still very good. The same can be also done for randomly migrating cells. So here you have a cell forming multiple fronts, and that is the raw data, and so this is the Meinhardt model, and so you see this splitting here of a front, okay, and then what I find surprising is so that even this fainter background shading is captured to some extent quite well, okay. Usually if you think about Turing patterns, you think about how can I create a very robust pattern, okay. And here we see so that even a highly dynamic pattern, so this spot dying out, can be captured quite well. The questions that arise here are, you have two different mathematical models, the Levchenko model, which also does a very good job, and the Meinhardt model. They are based on entirely different assumptions, but give you almost the same result, okay? We should discuss that rather over tea or a beer. I see I need to whiz through my last section, which is very much related to what Eva Palloch presented. So and she already said so that cells that move in confined conditions tend to turn to blabbing. So here we look at Dictyostelium cells that are seeded such that they chemotax sandwich between solid substrate, so glass here, okay, and an agarose overlay. And so you can see with increasing agarose concentration, so the number of blebs increases. And here you see a movie where here, for example, in that region, you should be seeing a prominent bleb. Yeah, there, for example, you see this very typical actin scar. So this is the old actin cortex that stays behind. And yeah, so these blebs, as we learned yesterday, they are myosin II dependent, and they're driven by hydrostatic pressure, and they're often found in cells that move in 3D constrained environments. And also what Eva alluded to yesterday is, so there is known regulators of bleb site selection, for example, weakening of the ectomyosin cortex, or local contraction of myosin II, or asymmetric distribution of membrane cortex linkers. So what I want to present here is so that one thing that has been overlooked previously is simply that cell geometry plays an important role via membrane tension. And so this is again very much based on imaging data, okay, so where we extract contours and then we have a method to actually extract events at the membrane. Okay, so here you see a time-space plot, so time going now from top to bottom, and here the normalized coordinate along the contour. And so you see different events, for example, here, what is that? It's a contraction at the rear in blue, okay, here, or you see protrusions, or you see very fast flaps. And so what became apparent very early on was that membrane curvature, which is plotted here, plays a significant role. For example, if we look here at this case of bleb number 1, so that emerges from a concave region here. Okay, so concave meaning here blue, and as it pops out, it creates negative curvature at its side. Okay, and then after some time, you see, so that is exactly from that region, so that a new bleb emerges. Okay, giving rise again to negative curvature on its side. And then you see many cases where you see whole chains of blebs where one is triggered by a previous one. I don't have the time to show too much of the data, so I want to directly jump to a conceptual model which we derived. So where in a convex configuration, so the hydrostatic pressure is balanced by cortical tension and membrane tension. It's actually very difficult to separate the two. Okay. If you look now at the concave case, so hydrostatic pressure is also directed outwards. But now, so are membrane and cortical tension. Okay. What we see, however, in the experiments is that the actin cortex always stays in place. So it must somehow be anchored in one way or the other, maybe to the substrate, or it's just that rigid. We don't know. So meaning so that the entire drag force resides here on the membrane, and the idea is now that the membrane is what gives way first. Okay, and this requires that any cortex membrane linkages simply break. Okay, and yeah, so you could say, okay, how can cells utilize that mechanism? Okay, and one way we think they do it is so they actually utilize their cyclic MP chemotactic machinery to create protrusions at the front, which then indirectly create regions of negative membrane curvature that then recruit blebs to the front, simply because blebs have a higher protrusive force than slow actin-based protrusions. And Eva already showed a very nice example of a fish cell that also moves by blebbing in a confined space, and we have analyzed that cell and see so that the very same principle is true here. So you see blebs and then new blebs almost always emerge from regions of negative curvature.
Do you identify boundary in the DIC images, or you have fluorescence just not showing it?
In here we only had that movie, but in Dictyostelium, so we used a technique where we use an indirect label. So we put fluorescent rhodamine dextrane into the medium, simply because you can't see the fresh black, it's void of actin. So if you have an actin marker, or we used a membrane marker, the cyclic AMP receptor, for example. But we're working on methods to segment from DSC images as well. So, yeah, this prompted us to come up with a biophysical model where we consider a membrane coupled to a cortex. Okay, so these linkers here are not drawn to scale, so their cortex should be much closer to the membrane. And then we can actively create a protrusion, for example, and then the negative curvature here in these two regions actually results in an outward directed force, yeah, where then the linkers will break. Okay, so we do not prescribe where the blebs are going to form. So this is simply dictated by the geometry. Okay, so here's a.
Excuse me.
Yes.
The viscous drag that you write in your model is the viscous drag of what? On what?
It's the viscous drag of the membrane moving through the medium. Okay.
So that's very low, right?
It's very low. Yes.
That's a membrane and water.
Yes. Yeah. It's mostly because of numerical reasons to make it easier to compute that. So we use a very similar approach to what I have shown before. So where we use a standard model for the membrane that resists stretching and bending. Okay, and we have some isotropic pressure, and then we have linkers modeled in the most simple way, so as linear springs that bring. Okay, thank you. I'm going to make that.
And why membrane resist bending?
It resists bend.
Yeah, even model membranes do that.
Yeah.
Just because you have to compress the lipids on one side compared to the other.
Or just compared to actin cortex, that would be miserable, right? Am I missing something? Membrane is pretty easy to bend by itself in the blood.
Yeah, I mean here it doesn't play such a prominent role in that model. Okay, yeah, it's more the membrane tension actually. So we then again take an approach where we use imaging data and try and predict from the imaging data where blebs are going to form. Okay, and here's a case where you see two blebs forming, and we say whenever there's significant enough overlap between the model bleb and the experimental bleb, we count this as true positive. Okay, so it's a bit iffy, but and one ingredient of the model that obviously plays an important role is the pressure, and so we optimize that for each individual cell and configuration, so that we actually optimize for the number of true positive blebs. Strikingly, you get a very nice result out of that, where you can see so that for two different agarose concentrations, we obtain a clear separation. Okay, so that the model predicts so that under 2% agarose, cells are under higher pressure, which is kind of a sanity check. And this corresponds to the roundness of cells. Okay, so more pressure the rounder the cells are. And
Some noise right
yeah.
there. How do you measure the pressure?
That is the pressure in the model. So this is not experimentally measured. Okay, so this is optimized in the model, but yes, of course, it would be great to have tools to measure that. Yeah, so I cut that short. So the idea is to now use a statistical approach, and then specifically look at cases where we make a wrong prediction. Okay, this is where we want to learn something about the biology, which is not captured in the model. And for example, we can say that half the number of false positives are due to some delay observed in the blabbing of daughter blebs. And here's an example. So here we have a bleb with a clear actin scar. The model would predict, because there's a highly concave region here, that a bleb pops out immediately. However, we only see that bleb emerging after a new cortex has been restored. And we can now measure from the images the actin build up in these blebs, and we try to incorporate that into a more dynamical model. Yeah, to summarize, so we can say, so membrane tension has been known to be able to quench protrusive activity at the cell rear, so working in terms of long-range inhibition. We show here so that tension has a dual role. So actually in concave regions, it can act as a local activator of protrusions in forms of blebs. And so this model, which is entirely based on geometry, is actually able to predict a large number of experimentally observed blebs. And that brings me to acknowledging a couple of people. So first of all, Sharon Molyneux, who is now working on the blabbing model. Robert Lockley did a lot on the cell porosity models, and Richard Tyson was instrumental in all the image analysis. And yeah, I thank you for your attention.
A specific question.
Yeah.
You mentioned negative curvature in this concave region. Is it really negative there?
It's a good point.
You're talking about mean curvature, which may be well positive.
As I have learned, so different communities use it in a very different way. Okay, and even in some papers, so some papers say, okay, a philopodium, the tip of a philopodium is highly negatively curved. Okay, and that is generally accepted in the biology community. If you look at some biophysics papers, so even in the very same paper, they later on change the notion when they go into the mathematical detail and define this as positive curvature.
But my question is very, very specific. What matters is the mean curvature. Is the mean curvature in the concave regions positive or negative in your specific case?
Here it's negative. I mean, it's a simple 2D contour. Yeah.
But the other direction bends around.
I mean, mean curvature.
You have two directions of curvature. One is what you see in the focus plane, and the other one is perpendicular to it. And in the end, both curvatures add up if you look at the mean curvature.
Yes, but here we only look at the slice. Okay.
Okay, so you ignore the thickness direction, basically.
Yes.
Okay.
Yeah, but in all
Because our cells are confined between two plates. Okay. And
So
it's
what
like a plank of wood where you cut something out. Okay.
But what is the profile of the cell in the third dimension? If you take a cross section,
It is flat. So you can
But
show.
right at the edge, the curvature must be very high.
yeah,
Because it has to bend around in that tiny gap.
it is even worse.
Yeah, it's even worse.
it gives
But
it
it's
even more
constrained.
curvature.
It's even
So
worse.
it's
You
constrained.
as Rudy just mentioned, by constraining the cell between two plates, you increase the second positive curvature. You have saddle-like shape, right? Saddle-like shape is characterized by one curvature which is negative, as you mentioned in the
Yes.
The second is positive. What matters in physics of membranes is always mean curvature, meaning the sum of the two.
Yes, but which will be different at the sides, obviously in the middle between the plates. Okay, as you said, so it's a saddle. Okay, so here we talk about the middle section. Okay.
I think in terms of the imaging, it's all right to do the 2D contour. I think what Misha is referring to is the restoring force of the membrane against such a curvature is dictated by the mean curvature and not just by the curvature in one direction. In the other direction, it's very highly curved.
Yes.
So the membrane may not want to bleb by your argument because it's curved in the other direction, and that may want to flatten it out.
Okay, fact is it blebs, so you can see it. Another fact is, so cells in buffer also show the same behavior.
Maybe the physics is different. Maybe the physics of this bleeding is not what is different from as compared to what you.
It could be. I'm very happy to discuss that. Yeah.
But
yeah, of
the
course.
geometric constraint can play a role. The fact that you're constraining 2D, it does play a role. It's not just a free membrane allowed to change its shape in 3 dimensions anymore.
Maybe physics is.
No more questions?
Maybe like in the model of Eva and Faluk and Guillaume, their like osmotic pressure was playing.
Yes, they have pressure gradients
because
on isotropic. Yeah, we ignore all that. Yeah.
yeah, because like the jump of osmotic pressure is higher of magnitude higher than the jump of hydrostatic pressure.
Doesn't matter.
So that
You're just
might be
finding the
one-
constant.
an important factor.
These
Yeah,
guys
yeah.
have higher curvature.
Okay, thank you.
Yeah, thank you very much.